great pleasure to be here. This is a picture of the Institute of Metabolic Science where I work, and there we kind of do we ask two questions. One is, why are some people fat and some people not? And the second question is, when you get obese, why do you get sick? And we try and use a wide range of technologies to address those problems and hopefully use that information to improve strategies for both prevention and treatment of metabolic disease. Now, I've used human genetics a lot in my work, and it's not unreasonable to ask, why bother? Why should we bother using human genetics? What's the point? It's a good question for any scientist to ask. And we generally bother to do things because they're either useful or they're interesting. And how might human genetics be useful? Well, people generally like having a diagnosis. If you can give people a specific diagnosis, it's much better than just giving them a vague terminology for their disorder. If we can make a specific molecular diagnosis, we kind of get a better idea of what's going to happen in the long term for people, and we can get improved prognostication. The great dream when human genetic revolution started is we'd be able to screen people early in life, identify those at risk of developing disease, and by selectively changing their behaviors or informing them, we'd reduce their risks. But that's still a little bit of a pipe dream. But what we have seen some definite advances in is the field of pharmacogenomics, where we can identify genetic disorders that can lead to therapies being targeted, at least selectively, towards individuals' particular disease mechanisms. Of course, we do science also because it's interesting, and genetics has taught us a lot about human evolution and development. I think one important thing that we sometimes don't remember quite as much is that genetics has been very useful in teaching us about human physiology. If we find people with, even rare individuals with genetic disruption of key molecules in controlling homeostatic feedback loops in the body, that can really give us an immediate understanding of how those loops are controlled. And not only is that interesting, then it can become useful, because those positions, those control nodes in the human genome can very rapidly turn into very useful therapeutically modifiable targets for novel drug development. A key thing about genetics, for those of you who don't think about it very often, is why do we do it so much? Well, it's really extraordinarily useful because it informs us about the direction of causality of our research. Many times, if you're doing research and you find two things varying, you're never sure which direction it is influencing what. Whereas if you find a genetic variant, it is very, very unlikely to be caused by the phenotype that you're studying, and it's likely to be unidirectional. And that's what gives genetics its tremendous power. So if we put together a picture of the key organs concerned with the control of energy balance, insulin sensitivity, insulin secretion, glucose homeostasis, this is the picture that we would see. The gut-brain axis, the liver and the muscle as key insulin target tissues for glucose metabolism. The adipose tissue is a storage organ, and the pancreas clearly as the insulin producing organ. And what I'm going to give you just this one slide to tell you broadly what genetics has really helped us learn over the last 20 years or 25 years or more. And in essence, it's as follows. When we're talking about obesity, a state of increased body fat mass, by far the dominant signal that we get from genetics is the brain. Obesity is largely a heritable neurobehavioral disorder with the variation between the resistant and the sensitive largely being based around genes expressed in the brain. And again, if I turn now to metabolic syndrome, insulin resistance, you might think that the targets that clear the most glucose, the liver and the muscle, might be the place where we'd find most of the action that determines who's insulin sensitive and who's not insulin sensitive. And rather remarkably, our only boring little tissue, fat, turns out to be hugely important in determining which individuals can cope with overnutrition and which individuals cannot cope with overnutrition. And then finally, when it comes to type 2 diabetes and actually developing a high blood glucose, the real determinant of who gets those high glucoses and who doesn't is is how well your pancreatic beta cell has been set up from birth. It's really a very dominant signal when you're actually looking for type 2 diabetes as your readout. So depending on which part of the whole spectrum you're looking at, you get totally different answers as to what's causing or what's driving metabolic disease. We started our interest in obesity some 30 or more years ago, and at that point, there had not been a single human described with genetic defects causing obesity, and we were fortunate to be the first to do so. And we and others, and my colleagues who started with me way back there as students and postdocs, have remained working in Cambridge, and we a highly collaborative group and team, Sadaf Baruki, Giles Yeo, Tony Coll. And what we found were genes, when they were disrupted, very reliably increase human body weight, sometimes very severely. And those genes were, in some ways, to some people, surprised. They were not in fat tissue. They weren't in tissues burning off energy. They were all in the area of the brain, largely in the hypothalamus, or in a gene like leptin acting on the hypothalamus. So we were able to show that genetic basis of highly penetrant obesity was all based around genetic disorders of the hypothalamus. We didn't suffice just to stop there and just describe where the genes were. We actually did take our patients in to the clinical research facility and measure whether they burnt off calories slowly or they ate more. And rather remarkably, the evidence was much more strong that these were disorders of appetite. So these so-called metabolic disorder of obesity was really not a metabolic disorder at all. It was a primary neurobehavioral disorder. You can see this in this graph here, where we look at kids with leptin deficiency, match controls here, kids with obesity due to totally inactive melanocortin 4 receptor mutations downstream of leptin or partially active. And this is remarkable. Here's a single molecule in the central nervous system, and depending on how much signaling deficit you have in it, you can predict how much people will eat when they sit down for an ad lib meal. So really these processes in our brain are intimately connected to really what is quite a sophisticated process, a decision about terminating a meal. And while some of these disorders are uncommon, melanocortin 4 receptors are not that uncommon. We recently did a birth cohort study. Every kid born in Bristol in 1997, in 1990. This is the remarkable ALSPAC study. And in that, we found that 1 in every 330 of those individuals had got a functionally impaired MC4R mutation. That means there's 200,000 carriers in the UK, 1 million in the US, more than 20 million worldwide. And on average, every one of those individuals on average went into adolescence 18 kilos heavier than they otherwise would have been. Here's a great example of precision medicine. It's rare, but it's beautiful and moving when you can be directly involved in it. And when we discovered children lacking in leptin, we're able to give back leptin to them. It was really a dramatic and totally revolutionary observation. These children, and now we can do it much quicker now than we did it then. We were very gentle with the recombinant leptin. Now we know it's very safe. And we can restore normal body weight within 6 months to a year in these children, some of whom are so enormous that they're almost wheelchair bound. And we learned a huge amount about leptin's physiology from replacing leptin. One of the nicer experiments we did with our colleagues in psychiatry, Paul Fletcher and Ed Bullmore, were these lovely experiments where we took some adolescents who had never seen leptin before and they volunteered to go in a functional MRI scan. And if you showed them pictures of things like cars or footballers, not much happened. But you showed pictures of food, and the areas of addiction and reward in the brain lit up like a beacon. Now, the remarkable thing is when you gave leptin back, you could totally restore the normal pattern in the brain just within days of way before they lost weight. So here's an extraordinary business of leptin, a hormone coming from your adipose tissue, powerfully influencing your higher brain centers concerned with decisions about eating and not eating. And similarly, now distal to the leptin receptor in the brain is the POMC system. And now we have agents that have a similar powerful effect on reducing body weight. Here's kids with POMC deficiency, work from Heiko Kruder in Germany and groups in Paris. And setmelanotide, this MC4R agonist, is now a approved NICE agent for children with defects in POMC or PCSK1, which we described many years ago, or leptin receptor. And there's ongoing and quite exciting phase 2 studies going on in things like Alstrom and Bardet-Biedl and other hypothalamic craniopharyngiomas, etc., where this is looking at like a very promising treatment for these conditions. So I'm going to move, this is a very quick talk, so a lightning talk really in 10 minutes. So I'm going to move from the causes of obesity to the consequences now. And obesity is really caused when energy intake exceeds energy expenditure. And that's really, obesity is an expanded mass of triglycerides in adipocytes. That's all it is. Sometimes the word obesity, I think, makes people stop thinking. They just think they know what it is. But really all it is the expanded mass of triglyceride in adipocytes. Now, why is that associated with such bad stuff? It's associated with things I classify into three. I think this is a useful way of thinking about it. There are kind of mechanical and gravitational effects where simply the weight and mass of the triglycerides are contributing to the disorders. But then there's cancers, which are associated, just these three, several, and a whole range of metabolic diseases. So it's pretty obvious that the obesity itself directly results in these. But why is it associated with cancer or metabolic syndrome? Is it the expanded mass? Or is it somehow that that's just a biomarker and it's chronically exceeding between energy intake and expenditure that somehow is directly causing these illnesses, with the obesity being simply an observable epiphenomenon in what's truly going on? And so there are kind of two ways of thinking about how you might get from this sustained energy balance. Let's say when it comes to insulin resistance, because these are the two key tissues for glucose handling, your liver and your muscle. So how do you get from there to there? If you ask 100 diabetologists these days, you'd probably, because they go to regular meetings, they've been listening to talks at meetings, and they'll have been told that the big bad fat cell is the culprit, that it stuffs full of fat, it gets damaged, it starts having macrophages, they produce adipocytokines, and it's these bad guys that are causing the problem in the insulin target tissues. The only problem there is that's true in mice and beautifully demonstrable in mice, but actually it's hardly supported by a single jot of data in humans. Neither human genetics nor human pharmacology support this idea very much. And whereas 20 years ago, every pharmaceutical company was trying to develop anti-TNF, anti-IL-6, anti-RBP4 to try and block insulin resistance, they all found that those strategies never worked. It didn't do a thing. I think that's because a different phenomenon is what's really going on. And it's that actually your big fat cell is not your enemy. Your big fat cell is your friend. Because the worst situation is when you can no longer take triglyceride and energy into your professional energy store, and then you redirect it to non-professional tissues. And it's the redirection of energy and nutrients and lipid to those non-professional amateur tissues, liver, muscle, endothelium, heart, where you really start running into trouble. And we've learned this really from both monogenic and polygenic studies. David Savage, my great long-term colleague, and I have discovered many of the genes that cause lipodystrophy. Lipodystrophy is a condition not of thinness. It's a condition when you cannot make enough fat cells, or you cannot make fat in fat cells. And it's either total, as in the patient on the left, or partial, as in this patient in the middle. And these people develop all the complications of obesity in the absence of excess stored triglyceride. There is not one feature of obesity that these people, or at least not one that isn't the mechanical and gravitational, but all the metabolic and the cancer risks, etc., are all the same in individuals with lipodystrophy. So years ago, I think this is one of the most important papers we've ever been involved with our colleagues in genetic epidemiology. We asked a simple question. We said, if we take metabolic syndrome as defined by high insulin, high triglycerides, low HDL, and we simply ask the genome in a totally hypothesis-free way, where are the genes in which genetic variants predispose to that cluster of insulin-resistant abnormalities? And we were shocked and excited, but shocked to see that the only tissue that came out was the adipocyte, and the only tissues was fat. So genetic variation that made people either sensitive or insensitive to metabolic syndrome was a very strongly adipose tissue dominated signal. And indeed, when you take that genetic signal, you take a polygenic risk score for metabolic syndrome, and you start bringing it into other data sets, like the data sets of people who have had 12,000 people with DEXAs, you start seeing remarkable data. If you have a score that gives you a high risk of metabolic syndrome, it's not so much that you have a lot of visceral fat. By far the most striking thing is that you have less fat on your hips and thighs. And so this absence of functional and absence of safe fat is just like a form fruste of lipodystrophy, is a common underpinning of the metabolic syndrome across populations. And so that led me to, this is the final slide, the one that I often use in public talks, and sort of puts everything together into the soggy bathroom floor model of metabolic disease. Let's, think about the fact you go into a cheap hotel, the bathroom is there, someone's left the plug out, someone's left the tap on, but the bathroom floor is fine because you get a store, you get a pressure, you get a continuous throughput, and everything's fine. Now, we normally think about metabolic disease as we are obese and we increase energy intake too much, and maybe we close up the tap and have decreased energy expenditure. And so we fill up the bath and then we get nasty, soggy bathroom floor. And that's the ectopic lipid and muscle that's causing the insulin resistance. You get compensatory hyperinsulinemia. Together, these lead to all the features of the metabolic syndrome. And then once we stop having the compensatory hyperinsulinemia with vulnerable beta cells, when that finally give up, then we get hyperglycemia. But what we forget is that not everybody has the same size bath. And I think our work has shown that this adipose tissue storage capacity is really a very important variable that's different between individuals on the basis of genetics, possibly of prenatal nutritional state. It's a place where you can integrate a lot of the insults that lead to the risks of developing later metabolic syndrome. I find this a useful model even in thinking about how we treat diabetes. And rather depressingly, we really treat it at the very end of its life with our drugs that increase insulin secretion and include GLP-1, part of GLP-1 actions in that really are very late treatmetments in this cycle. We don't really have any drugs that are insulin sensitizers of that we think that metformin and SGLT2 are, but they're really not. What they're doing is actually reducing the compensatory hyperinsulinemia by putting glucose into different places, SGLT2 into your urine, metformin into your gut and lining, and reducing the compensatory hyperinsulinemia and therefore taking stress off the beta cell and reducing some of the other risk factors. Physical activity does indeed improve insulin resistance in skeletal muscle. It's an important part of the therapy of diabetes. But of course, we've had the biggest advance through really going up to the top, and with our recent incretin-based therapeutics at last having access to the tap at the top of this particular problem. We can, of course, we do have access to agents that alter our adipose storage capacity, PPAR gamma agonists, thiazolidinediones. Unfortunately, they have some off-target effects which have limited their utility, but it's still, they prove the principle that if you can change the size of the bath, you can dramatically improve insulin sensitivity and reduce glucose levels. And there's huge activity going on both in our own lab and elsewhere in trying to improve the efficacies of these appetite suppressants, adipose tissue direct therapeutics, and ways of trying to get ectopic lipid out of muscle and liver. And I think the next few years you'll see a range of additional pharmaceutical agents coming that will help us target our therapies towards the particular abnormalities which are most dominant in some patients versus others. And I'll just finish by thanking, that's just it's a very old slide, lots and lots of people had over the years, our funders, but particularly our patients and participants without whom we would not have had a research program over the last few decades. So thank you all for your attention.